There are three main steps for urine formation. The first part is glomerular filtration, which we've talked about already. Blood is brought into the glomerulus, and then the pressure in the glomerulus pushes water and small soluble molecules out of the blood and into the glomerular capsule, which leads into the renal tubule. Filtration is not very selective. We shove good and bad molecules out of the blood and into the renal tubule. The next important step is tubular reabsorption. We need to reabsorb the good stuff. Anything that we want to keep, the nutrients like glucose and amino acids, we need to take back and not leave in the urine. There are also some additional waste molecules that we can put directly into the renal tubule in renal secretion. The final step is water conservation, saving enough water so we don't dehydrate by peeing away all of our water. We're going to focus for the next few minutes on tubular reabsorption, taking back the good stuff. There are a lot of solutes that get pushed out of the blood and into the renal tubule that never show up in the urine because they're valuable solutes that we want to keep, and we reabsorb them completely. Things like glucose and amino acids shouldn't show up in your urine because you should be able to reabsorb them and get them all back in your blood where you like them to be. Most of the reabsorption takes place in the proximal convoluted tubule. The proximal convoluted tubule, or PCT, reabsorbs 100% of the nutrients that are going to be reabsorbed, as well as almost all the bicarbonate, a little over half the water and sodium, and about half of the potassium and chlorine. That's a lot of good stuff all absorbed in one section of the nephron, just the PCT. There are two main mechanisms that we use for transporting molecules out of the PCT when we want to save them. Let me draw a section of the PCT on the board so I can show them more clearly. Here's a section of the PCT. The PCT is made of simple cuboidal epithelium, so a single layer of cube-shaped cells. And these cells are optimized for reabsorption because they have microvilli that increase their surface area. So here's a section of the PCT. The lumen of the PCT, where we find the filtrate, is in here. And anything we want to save, we have to be able to get out of the lumen and to the other side of the PCT cells, where it can be picked up by the peritubular capillaries. There are two main mechanisms for getting molecules out of the lumen of the PCT. The first one is what's called the paracellular route. In the paracellular route, water and very small molecules are actually able to wiggle out of the PCT between the epithelial cells. Because the cells of the PCT are quite close together, this only works for very small things. Something bigger like glucose or an amino acid will not be able to be reabsorbed by the paracellular route. But water can fit through, as can ions, things like sodium and potassium and chlorine. And also some small things that we would probably prefer not to reabsorb. Things like urea and uric acid are small enough to get out between the cells. As the filtrate travels through the renal tubule, we actually do reabsorb some waste. We can reabsorb some urea and some uric acid, but we're going to see where we can get rid of those again later. For bigger things like glucose and amino acids that can't squeeze out between the peritubular cells, they have to go through the cells. That's called the transcellular route. Molecules that can't fit between the cells take the transcellular route, where they have to go through the PCT cells. Now, since we're talking about water-soluble molecules, these are not molecules that are going to get into a cell very easily. We need to have specific transport molecules that can bind to the solutes in the lumen and transport them into the epithelial cell and then out again on the other side so they can diffuse into the peritubular capillaries. A lot of these transport molecules that are binding to the solutes are using secondary active transport mechanisms. They use the power of sodium. Sodium wants to go into cells because we keep the level of sodium very low inside cells. So we have transporters that will let sodium come into a cell and use that movement to power the movement of other solutes into that cell. So that's secondary active. This is important because since it's a form of active transport, it allows us to transport even very low concentrations of solutes out of the lumen and into the epithelial cells where we can save them. We also see quite a bit of reabsorption of water through this transcellular route. Some water is reabsorbed paracellularly, but a lot is also reabsorbed transcellularly. This is through the process of osmosis. In osmosis, water is attracted to areas of higher solute concentration. As we are actively reabsorbing solutes from the lumen of the PCT into these epithelial cells, the concentration of solutes in the epithelial cells is higher than it is in the PCT, and water will be attracted to that higher area of solute. Water is only able to get in and out of cells easily in the presence of transport channels, those being aquaporins. So there are aquaporins in the PCT that allow water to be reabsorbed through the transcellular route. Typically, we reabsorb all of the nutrients. We reabsorb all of the glucose, all of the amino acids, and all of the vitamins, because these are good things that we want to keep. However, transcellular reabsorption is limited by the number of transporters. There are only so many transport proteins available, and we can't reabsorb more solute than there are transporters available. So in some cases, you can have so much solute in the filtrate that you can't reabsorb it all. We see this in the case of diabetes mellitus. Diabetes mellitus is the typical type of diabetes we think about where blood sugar levels get too high. The blood sugar levels can get so high that there's so much glucose in the urine, it can't be completely reabsorbed. That leaves a lot of glucose in the urine. When there's more glucose in the urine, that attracts more water by osmosis, and we end up with more water in the urine. So we increase the urine output and the frequency of urination. Two of the main symptoms of diabetes mellitus that's not controlled are frequent urination and excessive thirst, because you get really thirsty to try and replace all of the fluids you lose through the excessive urination. In addition to tubular reabsorption, where we're taking back the good stuff, we also see some tubular secretion. In tubular secretion, waste molecules can be specifically transported into the renal tubule. There are a number of transporters that will transport waste directly into the renal tubule. That includes transporters for things like urea and uric acid and ammonia. These are important because these are small waste molecules that can escape from the renal tubule by the paracellular route, but then we can grab onto them and secrete them right back in using tubular secretion. We also see tubular secretion of some drugs, specifically the drugs that are natural sorts of products that humans have co-evolved with over many years. Things like morphine, aspirin, and penicillin are naturally occurring drugs, and we have transporters for those particular drugs that can grab onto them in our tissue fluids and actively transport them into the renal tubule so that they can be urinated away. We also get rid of some hormones this way. They're specifically transported into the renal tubule in order to get rid of them. In addition to simply taking back the good stuff or getting rid of wastes, tubular reabsorption and secretion plays an important role in regulation. There are a number of molecules, especially ions, like sodium, hydrogen ion, potassium, chlorine, calcium, a lot of ions that are regulated by how much we reabsorb or secrete. This is particularly important when we get to the idea of hydrogen ion. Why is hydrogen ion so important to regulate? pH. The level of hydrogen ion is the pH, and pH is vitally important to how the proteins are going to function. So we need to regulate pH. I want to take a few minutes to talk about the regulation of pH, or acid-base balance, because it's so important. Normal blood pH is about 7.4, somewhere from 7.35 to 7.45. The concentration of hydrogen ion in our blood is regulated by 3 different body systems. First, there's the circulatory system, which helps regulate the pH based on the buffers that are found in the blood. Second, we have the respiratory system, which we saw can regulate the pH based on how quickly you breathe. If you breathe faster and breathe out more carbon dioxide, then your pH goes up as the carbon dioxide goes up. If you hold your breath and retain more carbon dioxide in your blood, that brings your pH down. The final system that's important for regulating the blood pH is the urinary system. The urinary system can regulate the amount of hydrogen ion by controlling how much hydrogen ion is secreted into the renal tubule. If your blood pH is outside of the normal level, then you have some sort of an acid-base disorder. If your blood pH is too low, if it's below that 7.35, that's called acidosis. And there are 2 forms of acidosis. Respiratory acidosis is caused by an issue going on with the respiratory system, either hypoventilation, meaning not moving enough air in and out, or poor respiration. Perhaps plenty of air is moving in and out, but we're not exchanging gas across the respiratory membrane. So poor ventilation or poor respiration causes a buildup of carbon dioxide in the blood that decreases the pH and causes respiratory acidosis. There's also metabolic acidosis. Metabolic acidosis is caused by the activity of the tissues. So tissues producing too much acid result in a lowering of the pH that is not due to the respiratory system. That's metabolic acidosis. On the other hand, if blood pH gets too high, over 7.45, that situation is alkalosis. And as we saw with acidosis, you can have respiratory alkalosis or metabolic alkalosis. In respiratory alkalosis, the problem is hyperventilation, breathing out too much carbon dioxide. When you hyperventilate, breathe out your carbon dioxide, blood pH goes up. That's respiratory alkalosis. Metabolic alkalosis is actually relatively rare. There are a couple of things that can cause this. Either chronic vomiting, where you're throwing up a whole lot. What that does is it throws up your stomach acid, and so your body responds by taking a lot of hydrogen ions out of your blood and pumping them back into your stomach again. Chronic use of bicarbonate products, like certain types of antacids, can have the same effect. You neutralize the stomach acid, and the body responds by pumping more hydrogen ions into the stomach to acidify the stomach. That pulls them out of the blood and results in a higher blood pH. So that's what can cause metabolic alkalosis.